So I'm very happy to be present here in this webinar for this invitation. As Dr. Mon just said, I'll I come from DFT, so my background is strongly density functional theory, material science background. But I was asked today to present something slightly different, which is what we do a lot in my group in Paris right now, which is the study of transformation in general in computational material science. So with a very general perspectives, and the way to describe transformations, whatever they are, whether they are nucleations from solid phases from the liquid ones, phase transition, chemical reactions, so involving changes in electron structures, materials for energy, and so forth and so on. So hopefully, yes, if you can still see correctly my presentation. So what we study is generalized structural transformation in the broad condensed matter physics and chemistry. So as I mentioned, the phase transition under pressure, typically for extreme conditions, nucleation from the liquid phase, chemical reactions in solutions, and to do that, the main tool, as for I guess most people here, is ab initio calculations. In particular, since we study transformation, and so temperature is an important, at least temperature is a very important thermodynamic parameter, we do generally ab initio molecular dynamics or classical molecular dynamics. And in fact, the spirit of my presentation today is to provide you a general framework to study transformation, which are independent from the fact that they use classical or ab initio molecular dynamics, and is really the topology behind that it's important. So, and we will see that the problem for any transformation is the fact that we need to sample efficiently a 3n dimensional configurational space. And this is a big problem, a big mathematical problem that, by the way, we are trying to overcome lately by using also artificial intelligence and machine learning tools, as everybody else, I would say, in the world. And so our challenge number one is to find good collective variables which are capable to describe and project this 3n dimensional manifold onto the few relevant dimensions we're interested in. And then the second challenge will be eventually, if I have time at the end of my speech, to use data-driven approaches to improve the power of our prediction. And so I will take three very different examples precisely to show you that this kind of approaches are very general. So we'll focus on water, on materials for energy, and also, if I have time, in origins of life and chemical reactions. Okay, so let's start with water. And water is a nightmare. Water is the most important substance on Earth, a very precious one, as we know. But from the research point of view, is a nightmare because, as we can see in this slide here, the phase diagram is very complex. We have pressure in the axis and temperature in the ordinates and the y-axis. And we see that below, at a low temperature, below the liquid phase, we have several crystalline phases, but still below these several crystalline phases, which are thermodynamically stable, we have at least two or three amorphous phases, which is already an anomaly because in general, a system making glasses have one glass form. In the case of water, we have at least 2 or 3 different glass forms, which is very complex already. And the problem of this of studying from the experimental point of view these glass forms, which are also very important in planetology because these amorphous ices are very much found in comets and planets and cold planets. The problem is that in general, for glassy systems, we have a direct connection from the liquid, so you are quickly cooling the liquid and you end up in a glassy form. This is not possible in water because no matter how quickly or slowly you cool water, you always end up in a crystalline phase. So to make the glassy forms, you have to do something very different. And this the right-hand side of my slide shows that actually there's a strong hypothesis. You see below here in the bottom part, you see that there's a HDA, high-density amorphous, and LDA, low-density amorphous forms that are separated by a continuous first-order phase transition line, supposedly terminating into this C2 red dot, which is a critical point, which has never been observed experimentally because it's a part of the phase diagram which is non-reachable from the experimental side. So this is just a little example of several of the water anomalies that have been at the origin of more than 50 or 60 years of intense studies, and we are not over the hump yet. But something much simpler that everybody can easily understand is just the study of the nucleation of ice, hexagonal ice from liquid water. Precisely what you would obtain if you put a glass of water in your freezer, you wait a little bit, and after I don't know, 20 minutes or so, you find ice. So if you look at the phase diagram of water, this is precisely when we are around our ambient pressure and about 300 to 273 precisely Kelvin of temperature. When we do simulations, it is very easy to melt, to start from the crystalline ice, as you see in the left part of the screen, and to melt it to form a liquid. But to do the other way around is very difficult. It's something that you can never obtain just as easily if you just take your liquid water, whatever you do classical simulation, ab initio simulations. If you take your liquid water and just and hope to find, no matter how big your simulation box is, you hope to find crystalline ice, you'll never get it. There's no way you'll ever get it. Why? Even if ice is more stable from the thermodynamic point of view at low temperature, you'll never obtain because you are kinetically trapped. So typically people, to try to freeze water in simulation, use some trick. They use some ice sheets to favor the nucleation, to already have a seed of the ice nucleus. There's a famous paper from Valeria Molinero about 15 years ago, a Nature paper, which was the first one where a direct nucleation of the water ice from liquid water was observed in simulation, but at the price of a very simplified interaction potential. And other tricks are used. Typically, you have to use a seed, an ice seed, to make ice nucleate and grow, starting from this initial seed. But if you want just to take just pure water, bulk water, and just wait, you'll never get it. And the reason, as I mentioned, is kinetical, because even if it's pretty much like you have a golf course, you have several holes which are much favored from the thermodynamic point of view. In a golf course, of course, is the gravitational potential that makes the ball want to drop into the hole. But the problem is that this hole is very narrow and the landscape can be very big. And so it's very difficult in your simulation to happen to be precisely around the thermodynamic hole constituted by the crystalline phases. So the problem in studying transformation from disordered phases to ordered phases, or from one ordered phase to another ordered phase, is that you have to be sure to locate the thermodynamic minimum to be able to go into that. And that's a very big problem. And that's actually the kind of problem that we started to study in my group in Paris from a very general perspective. That is, we ask ourselves this question: is there a general way to describe and simulate many different transformation mechanisms? And as a transformation mechanism can be just, for example, the graphite to diamond phase transition that you see here, two crystalline phases of carbon. The carbon atoms are exactly the same. They are arranged differently in because of their chemistry, from an sp2 to an sp3 configuration, but the studying, describing from the atomistic point of view this transformation is far from being easy. or you can have, as I mentioned, the nucleation of a solid crystalline phase, for example, ice starting from liquid water. You can have other very different problems, like protein folding is also another transformation occurring in the configurational space of proteins. Or you can have chemical reactions in solution where the atoms from the reactants and the products at the end of the reaction are exactly the same, but they are arranged differently. And this different arrangement is very, very difficult to be tracked in computer simulations, no matter what, no matter if they are a classical or ab initio simulation. Then this is something that probably you know, but just worth mentioning. When we try to sample a configurational space and a phase space, basically we do computer molecular dynamics. Molecular dynamics, you move atoms and you just solve numerically the Newton equation for the atoms, while the interaction, the forces here can be calculated either from a simple model, an analytical model in classical molecular dynamics, or via DFT ab initio calculation if you want quantum molecular dynamics. This is a skip. And then the important thing is that you have a connection between ab initio molecular dynamics and statistical mechanics. That is, if you have a generic observable, which can be the energy, the force, density, whatever general observable S, depending on all the atomic coordinates of the atoms in your system, you can track over time the value of this observable S. So you have a trajectory of S, a function of t. If your simulation is long enough, so if you are ergodic, then you can calculate the probability of the presence of the values of S throughout your simulation. So you're making a connection going from the time average to the ensemble average, which is at the basis of the ergodic theorem. And then, again, if you are statistically convergent, you are allowed to do this. From the probability, you can get the free energy, which is the important thermodynamic quantity, because the free energy tells you whether one configuration is a favor with respect to other one. For example, here in this simple example, you see that the value of S corresponding to the largest peak of probability P corresponds also to the lower minimum of the free energy. Then there's a second peak, which has a slightly higher minimum in the free energy, a very narrow peak here, a shallow peak here, which corresponds to a shallower minimum in the free energy, and everything else you have barriers, because there are states which are not clearly not favorable for this value of S. So when we do molecular dynamics, whether it is classical molecular dynamics or ab initio molecular dynamics, at the end of your as long as possible trajectories, you want to get the free energy taking as starting from the probability of presence of the observables you are interested in. And that is very difficult because, for example, if you want to study this is typically a problem in chemical reactions, because in chemical reaction you start from reactants, but then you have a plethora of possible products, and in principle you'd like to study all the possibilities, all the chemical network starting from given reactants. But then again, you are confronted to the fact, as we see here also in this phase transition from graphite to diamond, that you all know very well the local configuration of each atom in the graphite, because each atom is an identical environment with respect to the others. The same is for in the diamond in the final product in the diamond phase. What is very difficult to study whatever happens in between, which is the interesting part, because when you have a transformation, so a phase transition, and in this case, what you're really interested in is how the system goes from one phase to the other, and how difficult it is to go through this barrier. So how costly this is from the thermodynamic point of view, and how often this will happen from the kinetic point of view. So any transformation, for any transformation, we are interested in general in the thermodynamics, so the respective equilibria, and the kinetics how much time does my system need to go from phase A to phase B, or from graphite to diamond. And to have a atomistic point of view, atomistic calculation of whatever happens in between, you have to be able to describe this configurational space, which happen to be 3n-dimensional, because you have n atom in your unit cell. You have 3 spatial coordinates per atom, so you have your configurational space is 3n-dimensional. And when you go from graphite to diamond, in principle, your transformation occurs in this 3n-dimensional configurational space. And this is precisely the space we want to track, to be able to correctly find the transition, the lowest energy, free energy transition pathway, which corresponds to what physically the system will do, and so to, for example, to the kinetic barrier and the time necessary for the system to go from graphite to diamond. This is a very pertinent example, because graphite and diamond are almost equivalent in free energy, but you cannot. It's not very easy to take the graphite from your pencils to transform it into diamond, even if their energies is the same, because the barrier, the kinetic barrier, is huge, and so you'll never get it by hands. Otherwise, everybody would have diamonds out of pencils. And to do that, you need very, very high temperature and pressure, precisely to allow these graphite carbon atoms here to overcome a huge barrier and then end up in the diamond phase. So, and this is precisely the kind of problem that we like to study, because if we have an A system and a B system, we know that we want our system to go from A to B, because B is interesting, B is more favorable. Still, which is the pathway atomistic from the microscopic point of view that the system A will undergo to end up into B? This, of course, will depend not only on A and B, but also on the external conditions, temperature, pressure, and many other factors. So the system could go directly from A to B, could go through an intermediate C, through another intermediate D, etc. So this is the problem of any transformation in physics and chemistry. And from our point of view of simulation, the problem is a timescale problem. Because in general, if, as you see in this sketch, we have the A, which is the reactance, and B is the product. So we have a free energy, a reaction coordinate that allows us to go from A to B. In general, for example, I'll show an example in a moment. The free energy barrier is much larger than the thermal energy. So this means that we have a time scale problem, because the time necessary for the system A to overcome this barrier, this delta G star barrier, just out of thermal energy, depends exponentially on the height of this barrier. So if this barrier is much larger than KT, basically you'll never be able to observe it. And this is true because, for example, if you take a 1 electron volt barrier, which is very low, it's a typical physics barrier, but for example, chemical reactions typically have barriers of 30 kilocalories per mole. This corresponds to 1 year of actual time of simulation, or 1 day, if it's just 1 electron volt, of actual time of simulation. And of course, it is several orders of magnitude larger, not only than the FT, but also of classical molecular dynamics. So the very big problem for us computational material scientists in studying transformations is to overcome, to be able to sample transformation having a barrier which is much larger than the typical time of time scale of our simulations. So there are several ways nowadays to overcome this problem. One of the most famous, which was suggested about 20 years ago by Alessandro Lai and Michele Parinello, is metadynamics. In metadynamics, basically you run your typical molecular dynamics. Again, it could be classical molecular dynamics or ab initio molecular dynamics, and then you still have your observable S that I mentioned before. Every once in a while, you drop a Gaussian of energy like literally you are dropping some sand in your hole, and then little by little you have an infinite reservoir of sands. Little by little the sands will fill up your hole, and this will allow you to overcome the barrier and to find another minimum, and little by little fill up the second minimum. And when you are at convergence, your free energy pathway will be completely flat, and you will know the free energy landscape precisely because you will know where and when, how much energy, our sand or our water, you have put for each value of your observable S. So in the end, this blue part here will be precisely the free energy landscape you were looking for. The problem is this is something that is very robust. It's been 20 years now, so now people are very confident with this. The problem is, okay, but how do we choose this collective variable? That is, you're going from graphite to diamond. You have n atoms in your unit cell, so 3n special coordinates. How do you choose one variable that collectively describe this transformation of sp2 carbon atoms in graphite into sp3 carbon atoms in diamond? Of course, for simple transformation, you can take human descriptors such as distances, angles, dihedral angle, coordination number, density, energy, whatever. But in general, this can be easy for graphite, because in graphite, you know, they are going from a 3 coordination to a 4 coordination. So the coordination number of carbon atoms could be a good collective variable. But for other systems, it's much less easy to find the best possible, the best collective variable is the one that minimizes the free energy throughout the pathway. So going back to our nucleation of ice from the liquid, again, we are in our golf course, and we hope to find a way to force the system to get into this minimum corresponding to the crystalline ice, which is at low temperature, of course, favored with respect to liquid water. And so what we do is we introduce this topological variable, topological collective variable, which is called the permutant invariant vector, PIV, which we published a few years ago on a PRL. And the idea is very simple. So you have your, you take just, for example, 2 water molecules. Of course, you can generalize it to hundreds of water molecules. And so you create a graph out of you enumerate these, give numbers, labels to this, to each atom, the oxygen atoms and the hydrogen atom on each water molecules. And then you create a distance matrix. For example, the distance between 1 and 2, which are the 2 oxygen atoms, is 3.5 in angstrom. The distance from 1 to 3, which is its intramolecular hydrogen atom, so 1 to 3, it's 0.96, which is the intramolecular distance. While the distance from 1 to 5 is the H-bond distance, and it's about 2.5 angstrom. So you build this distance matrix, which of course depends on the system, on the pressure, on the temperature, and then you use a switching function that transforms this distance into values comprised between 0 and 1. So which we call the adjacency matrix. And then for if you have n water molecule, you are filling a half matrix of these distances, you transform this matrix into a vector, and then you sort this vector according to the values. So you put here, you have only one oxygen switching function distance, which is the only value here. You have 6 light blue distances, and they are sorted in order, and then you have the same into the violet hydrogen distance, and again they're sorted like this. Oh, and another example here of the same thing. And once you have transformed this distance matrix into a vector, into a sorted vector, and the sorting is very important because this is allows to be with respect to permutation. That's why we call it permutant invariant vector. Then once you have a vector, you have a matrix, because you can calculate, you can take one configuration, you have one PIV, you take a second configuration, you sort the vector precisely as we do in the first configuration, and then you can calculate that the Cartesian distance between the first vector, that is the first configuration, and the second vector, that is the second configuration. And this is something very, very powerful, because we are basically projecting all the coordinates of the water molecules in our system into a single number, this distance, which is a purely Cartesian distance of a space, which has, however, n times n minus 1 divided by 2 dimensions. So it's a very, very long vector, but the distance between two vectors, the Cartesian distance, is a single number. So we managed to project all the n water coordinates into one single number, and so we can use this single number to do our trick. So we play our metadynamics, and so to go from the water configuration we have here on the left hand side to the ice order configuration we have here on the right hand side. So we can do then this metadynamic transformation, because we have defined our good collective variables, this distance is defined, and so this allows us to converge, to obtain for the first time, that's why we published in PRL, to obtain for the first time water ice just from pure bulk water. So we are in this part of the phase diagram. So we are at, of course, at about room temperature and 1 bar. So we are perfectly timing conditions. So again, we are trying to go from liquid water to ice, to hexagonal ice. So this is a snapshot of our simulation, and on the low left part of the screen, you can see the little dot correspond to the actual free energy. So we have to go to the ice side, which has a lower free energy, but we have to overcome a barrier. So that's why we need to use metadynamics. And so you can see that little by little we push once we have defined this good collective variable S, we push our system away from the liquid phase. At some point we arrive at our transition state, where the system is half liquid, half hexagonal ice, and then the system can easily fall into the fully crystallized ice one. And again, this is done at ambient conditions, so we didn't need to supercool our system. The important part was to find the good collective variable to connect the liquid in an easy way, in one dimension, the liquid form with respect to the crystalline form. So again, we played around showing that we could do for this model of water, the melting temperature is slightly lower than the experimental one, so it's 250 Kelvin. So we could go from water to ice at 240, so 10 Kelvin below the model melting temperature. So we go from the liquid to ice here. But we also could do that, which is quite incredible for this kind of simulation, also at higher temperature, higher than the melting temperature. Of course, in this case, the transformation from liquid to ice is uphill, and the ice basin here on the right-hand side is very, very shallow, and the system, of course, will revert, if left alone, will revert immediately into the liquid form. So we played with a high pressure transformation of ice. I spare this for you because it's very technical and we don't learn much more. And so this is one first example on how to play with collective variables to try to study in a very efficient way transformation. I'll go very quickly on the materials for energy side, mostly to tell you something that is going on, but not fully achieved yet. We have in Paris, we have a consortium called MatSQ, which is an acronym for Materials for Energy through Stochastic Sampling High Performance Computing, involving several people, several postdocs and PhD. And the idea is precisely to study this kind of problem. We have a material for energy. In general, you have a material which has two states, a storage state and a release state. For example, a battery, you have a configuration in which you are storing the energy as chemical energy, and then a configuration you are releasing the energy via electrical energy. And so your system, which whether it is a chemical system or whatever, undergoes typically a reversible transition from A to B and then from B to A. This is the principle of every energy storage material. So studying these energy storage materials, of course, and predicting, designing new materials, of course, requires to be able to study transformation. That's why we are very much interested in transformation. So the principle is the same. We want to describe reversible phase changes, which are typically collective and out of equilibrium phenomena. Again, here we starting from the Schrödinger equation, we want to apply to a plethora of different possible transformation. And the bottleneck, as I mentioned, is again the fact that we have a 3n dimensional space. So in this project, we are working with mathematicians and computer scientists to find a mathematical way to choose the best possible transformation coordinates starting from our 3n dimensional space. So one of the main goals of the consortium is to optimize this dimensionality reduction and the coordinate optimization, while the second part is more application driven to study basically the interfaces of batteries and supercapacitors. So the first part is technical. I'm not deeply involved in it. It's just to quickly to tell you to go farther than what I just mentioned, to go from these two states and to define the generalized Langevin equation, be able to map this problem. Again, this is very mathematical, so I skip it because I'm not personally involved in this part of the project. The second part is to study materials for energy. And again, this is a typical problem where transformations are essential. So this is a sketch of what happens at the interface, at the water electrolyte-electrode interface of your batteries, the ones you have in any of your electrical devices. So you have graphite electrodes here, for example, and then you have your electrolytic solution, typically lithium with some complicated anions here, like PF6 minus or something else. Problem is that differently from what one may imagine, there's not just the electrodes and the electrolyte solution, but once you switch the electric field, so you switch the electrical part, there's this solid electrolyte interface, SEI, which is formed, which is typically 30 to 50 nanometers thick. And this part here is very crucial to stabilize the problem. If we didn't have this SEI, the batteries would just not work. So it's important because otherwise the system will break down. But on the other hand, this can, of course, since we don't know what's exactly in this interface and how it forms, this is a problem for batteries, in particular for the lifetime of batteries. So learn what happens at this interface. We have several crystalline phases. You can see in the picture: lithium chloride, lithium fluoride, lithium oxide, polyolefins. You have several different phases forming together, encrusting together over the electrodes, and we know nothing on how and why this form. So again, this is one of the main goals of our project. Is the start of PhD thesis, which will begin in next October, is precisely to model these supercapacitors and to try to understand with our topological definition of transformation how this interface forms, which phases are formed under which condition of salinity of electrical field and so forth and so on. So this is a nice sketch of the problem we're studying, and it's also these systems are huge, so we need to have a very efficient way to do this. And so here we are mixing ab initio part to study reaction, classical part to study the electrolyte in between, and then machine learning parts to make the joint between the ab initio part and the classical part. Let's skip the detail because I don't have too much time. And then I want to focus on something completely different that you might think being very off with respect to what I just said, that is the study of origins of life. The study of origins of life, of course, I will not go through the whole history of the origins of life, but modern study of the origins of life arrived when Darwin, about 150 years ago, made the famous assumption of the warm little pond in which ammonia, salts, water, electricity, pressure would do something to make life happen and then evolve into life as we know it today. And something that is a little less known from the general public is that in the 1920s, so about 100 years ago, there was two people, Hopper and Haldane, who formulated a hypothesis of a chemical evolution. Darwin was a biological evolution. Hopper and Haldane suggests that water, ammonia, methane, hydrogen cyanide, and carbon monoxide, like shown here, could make the bricks of life, proteins and nucleic acid or everything else, just as a form of evolution, so aggregating together, doing larger molecules, which would aggregate together, make larger molecules, in a really sort of non-living evolution, purely chemical evolution. And so in the early 50s, there was Harold Urey on the low part of the screen, who was already a Nobel Prize in chemistry, and his PhD student, Stanley Miller, in the upper part of the screen. And the PhD work of Stanley Miller was to take the 1950s hypothesis on the early Earth atmosphere, which were strong volcanic activities, lightning, and reducing chemical composition, and recreate this early Earth condition in a laboratory like this. There should be a sketch here. Hopefully work. Yes, you have a flask of boiling water, which represents the evaporation from the oceans. This water vapor goes into the atmosphere, which contains some simple gases shown here. And then they put an electrical discharge to mimic the effect of lightning. So they are reproducing the atmosphere of the Earth with the lightning in a box. And then via this cold trap, the system was cycled over for about a week. And in the end, the analysis of the here showed that there were a lot of organic compounds, including many amino acids, including glycine. This was a huge moment, because it was a moment for the first time it was demonstrated that simple molecules could aggregate and make amino acids, which are the simplest bricks of proteins, and so the simplest bricks of life. But the problem is, okay, this is nice, but how did this occur in the early Earth, where there were volcanoes and acid lakes and hydrothermal sources? I mean, a plethora of very different environmental conditions. And to give you an idea, so studying chemical reactions, these simple chemical reactions, for example, making amino acids, is very complicated, because studying a reaction means to study the equilibrium of the reaction, which is the thermodynamics, and the kinetic rate, the rate of the reaction. So, but to do this, you are confronted to a very complex problem, because the reaction did not occur in gas, in isolated system, but they occur on the Earth. In the Earth means that there could be there could have been atmospheric synthesis of bricks of life, so precisely as the example, the Miller experiment that I showed you earlier, something occurring in the atmosphere, for example, because of electric discharge provided by lightning. It could have been extraterrestrial delivery, so these molecules would form in space and then they would be brought down to Earth by meteorites and comets, or this reaction could have occurred in the Earth in aqueous phase on the interface of minerals, or in deep underwater hydrothermal conditions. In any case, studying chemical reactions in this context is very, very complex, because you have not only have the reactants and the products, but you have all the environment: water, salt, temperature, pressure, pH, salinity, electrical discharge, minerals, very, very complex. And that's why this problem is far from being solved. And so, if we want to, what we did a little less than 10 years ago, a colleague of mine was to study, to simulate this Miller experiment in ab initio, doing ab initio molecular dynamics. The problem is that to study origins of life with ab initio molecular dynamics, you have to use DFT to, of course, calculate energies and forces, then to do molecular dynamics, or ab initio molecular dynamics, to move the atoms to explore the chemical space. But then, as I mentioned, to go all the way, you will need to find all the possible reactions, all the possible reaction mechanisms, and accurately determine the thermodynamics, that is the free energy, and the kinetics, that is the rates, how often the system overcomes the kinetic barrier, to make this to see how often, how likely the synthesis, the abiotic synthesis of these biomolecules could have been possible in the early era. So to start from this, we started to just trying to reproduce the Miller experiment, that is starting with simple molecules: water, ammonia, methane, nitrogen, and carbon monoxide, and try, again, this is a sketch of the Miller experiment, and try to make glycine, which is the simplest possible amino acid. And I'll skip, yeah, I probably have a couple of movies here. Let me just see if I can start it. Yes, so you can imagine, you have to imagine there's a full simulation box that you don't see, because all the other molecules are hidden. But you might have seen there's this formation of this key molecule, which is called formamide. I'll skip all of this and just show you that at the end, after several long simulations with an external electric field capable to mimic a little bit the electric discharge, we ended up making glycine. And this was a nice paper we published about 9 years ago. For the first time, we reproduced the glycine synthesis under electric field, so a shortcut from the original Miller experiment. But the question was that we did it by making a chemical process which is different from the process that was supposed for about 60 years, which was called the Strecker process. So just to tell you that when we published this paper was a crazy media buzz. We were on all the papers on the earth for 1 month, because clearly it was the first time ever that computer simulations, ab initio computer simulation, were used to study the very earliest steps of the prebiotic synthesis of an amino acid. But again, the problem to go from the proof of concept to the quantitative study is again to find a good collective variable. And chemical reactions are again a very, very nice, difficult and nice playground to understand this concept of configurational space. Let's take this case. So I mentioned earlier that at some point, one crucial intermediate was this guy, numbered 1 here, the formamide guy, where you have carbon in gray, oxygen in red, nitrogen in blue, and then 3 hydrogen atoms. Suppose you want to break this formamide into carbon monoxide and ammonia at the right-hand side. If you do that in the gas phase, it's very easy. Basically, you have to break this carbon-nitrogen bond, and to have this nitrogen bonded to the carbon go to the nitrogen atom to make ammonia. And that's very easy. So it's not, you can do it literally by hand. But if you want to do this, so going from formamide to carbon monoxide and ammonia, but if you do that in water, but the problem starts to be different, because you not only have your 6 atoms of the reactants and product, but you have all the other water molecules around. And not only the other water molecules play a role, an indirect role, if they just favor or not as a solvent the reaction. The problem is that water also has hydrogen atoms, and you don't know whether the final hydrogen atoms here in the ammonia is the original one that was bonded on the carbon, or some of the other many hydrogen atoms present in the solution attached to water molecules. How do you know that? You don't. So again, this is a typical case in which you have to devise a topological scheme capable to project your 3n-dimensional space onto a few, 1 or 2 reaction coordinates. And this is precisely what we did with my colleague Fabio Petrucci in a paper we published in 2015. We take formamide here, carbon monoxide and ammonia here, and we make a matrix of connection. That is, the carbon atom here is connected to 1 oxygen, 1 nitrogen, and 1 hydrogen on the left-hand side in formamide, but only 1 oxygen, but 0 in nitrogen and hydrogen right-hand side. Oxygen doesn't change, it's bonded to 1 carbon on the left and 1 carbon on the right, so all the numbers are unchanged. Nitrogen also has 1 carbon on the left, but 0 on the right, 2 hydrogens on the left, 3 on the right. So basically, this 4 coordination number, we make a combination of these 4 coordination number, we make them continuous, so we go continuously from 1 to 0 or from 2 to 3, and we create a collective variables which is given by this combination of coordination numbers. And then again, we play our meta-dynamics. So here I can show a little movie where you have your cursor here that gives you the position in this projected space. The S coordinate, when you have 1, you have formamide here. When you have 2, you have hydrogen cyanide and water. So the system stays in 1, and then the color is the free energy. So you do your meta-dynamics, you're creating a free energy hole in your system. And then at some point, the system will switch on the right-hand side here, and you see that the molecule is now the hydrogen cyanide, the linear hydrogen cyanide molecule, and throughout the process, you have created your full free energy landscape. So this is an approach that we have used several times for several publications, several good quality publications, as you can see here. We had a collaboration from NASA on this part, because something that is very a big question for the Astrobiology Institute of NASA is why all the amino acids are left-handed in living system on the Earth, but they are racemic, half left-handed, half right-handed in space. So you take an amino acid in space, it can be left or right-handed. If you take any amino acid out of a living system on the Earth, it's always left-handed. Why? Where does this difference up? So I skip you the detail, just to tell you that we focused on 2 particular amino acids, and then in the end we found that there was, again, with our method, we could create the free energy, make the free energy calculation for the deamination or decarboxylation of glycine, and then the same, for isovalene. Just to tell you that our method, for our topological based method, allows us to study any reaction in solution, and so to calculate the free energy and the kinetics of any reaction, taking into account all the solvent, fully ab initio, fully DFT, without any other tricks, and finding the best possible reaction pathway. This is just another application to the synthesis of the RNA nucleotide. At some point, again, we are playing with our tricks. At some point, you'll see that this in the middle, that these glycosidic bonds that form between the ribols and the adenine, so we're making adenine monophosphate, which is one of the building blocks of RNA. And the final part, I don't know if I have time. Hello?
Yes. Yeah, you can continue. Maybe 5
so
more minutes.
just to finish, all of this, in particular, this chemical reaction part is done fully ab initio with a very smart, I think, use of the projection of the coordination, the highly dimensional coordination space, chemical space, onto some very small number of coordinates. But still, this kind of calculation for chemical reaction is very, very cumbersome, because to convert the free energy, it takes forever. It takes forever for ab initio calculation. So recently, we started to use machine learning. Dr. Moni told me that you have seen several seminars on machine learning, so to avoid it a little bit, but so that's why I only speak about machine learning only at the very end. We know, you all know, that ab initio calculations are expensive. They go as n to the power 3, and then if you do more advanced methods like quantum Monte Carlo, the scaling is even more unfavorable. And also we have this our ab initio trajectories, which are on the nanosecond time scale at the best. Sorry, this is wrong, nothing to do here. So the idea is, okay, can we, of course, replace some of this costly ab initio calculation with machine learning? I'm sure that you have seen many of these, so I'll go very quickly on this part. You know that through, for example, through neural networks, you can learn by training to recognize a cat from a dog or a handwritten number, and the same can be done, of course, for computer simulation, for ab initio computer simulation, because if you provide your system a large number of trajectories that is configurations and energies corresponding for picture of cats and dogs and the label, this is a cat, this is a dog, we can do the same using appropriate descriptors and etc. for ab initio calculation, and that's what we have been doing in the last few couple of years. So the idea is, of course, again, I think that everybody here knows what neural network potential can do in reproducing ab initio calculation without doing the ab initio calculation part. Of course, the nice part is that with respect to classical potential, in the case of neural network potential, you don't need to choose a particular mathematical form of your potential. You can have a local perspective and so split the total energy of the system as a sum of individual atomic energies, precisely as is done, for example, in the SOAP approach or in the Beller-Parinello approach. And again, going back for our multi-scale simulation, is to be able to do simulation at a higher level, at a more macroscopic level, but still keeping the quantum precision, quantum accuracy. And again, if you remember what I just said on prebiotic chemical reactions occurring in solutions with minerals, salts, pH, temperature, pressure, whatever, I mean, you can never think of doing this properly with ab initio calculation. In particular, if you want to study to sample correctly the space to calculate the free energy profiles, it's impossible, just impossible. So the idea is to try to do as much as possible ab initio calculation. So we took another study that he didn't have to mention, the Strecker reaction, to make glycine differently from what I show you today, to take 2 of these 7 chemical steps and to try to reproduce this using machine learning. The problem is that when you calculate the free energy, you have to do that over splitting your reaction coordinate into like 40 different windows, and for each window you are supposed to calculate to do your ab initio calculation, and this is very costly. So what we tried to do was to have a single machine learning model, but train not just on the reactants on the left-hand side or the products on the right-hand side, but also on the transition state or two other points of the transition state. Again, I'll make the story short, but what we observed is that for different kind of reactions, just using from 3 to 8 training points, we could reproduce the full free energy landscape, which taking basically 1 order of magnitude less of ab initio calculation. Again, I keep this very short because I was told not to talk too much about machine learning, just to tell you that we are studying other transformation, for example, the liquid phase transition of sulfur, which was discovered in a Nature paper 3 years ago, to try to study using machine learning, because again, it's a very complicated problem. I'll skip this part. And so I come to my conclusion. So what we study in Paris are generalized structural transformation, again, phase transition, nucleation from the liquid phase, chemical reaction in solution. There's a colleague of mine who also studied proteins. I show you 3 emblematic cases: the liquid ice nucleation, the origins of life, and I just mentioned some transformation in batteries. In principle, we do ab initio molecular dynamics, but the bottleneck being the efficient sampling of the configurational space, we have done some kind of work to define and robust, good collective variable to describe this transformation, and later on we are also using machine learning approaches. So I thank my collaboration. I thank Dr. Moni and all the virtual lab company for this invitation, and I'm open to questions.